Aquaculture video lecture. This is for the Friday before March 22nd. We this and I'm just going to put up by a video. And here is where we left off. We had some case studies. And the last case study that I wanted to talk about, remember we talked about already the Exxon Valdez oil spill, and then also the oil spill that was associated with the Iraq War in Kuwait. But here is the third and probably most disastrous oil spill was the Deepwater Horizon oil spill that happened in 2010. I'm not going to attempt to show this video directly through this video lecture. I just don't think it will work, but I really would like for you to watch the Deepwater Horizon video. I also recommend the movie. It's really good. And it shows you, it's a little more dramatic than what happened in real life, but it's what happened. It was definitely overambition on the part of British Petroleum. British Petroleum owned the drilling platform called the Deepwater Horizon because it was brought online early prior to sufficient testing. It did explode. Yeah, it was brought online early in order to meet the demand for oil, and it released 600 million liters of oil into the Gulf of Mexico. So it was about, I want to say, about 6 times larger than the Exxon Valdez spill. Okay. And bioremediation was very aggressive in cleaning up this particular oil spill and accounts for about 50% of the oil released was bioremediated. Okay. And so the plume of oil was actually underwater. It was crude oil can be more dense than water, can be less dense than water. But the plume itself had a density that was very close to salt water. And so it was an underwater plume. It was not on the ocean floor. It was just kind of trapped below layers of water. It was huge. It was about 22 miles long. There are microbes already in marine environments that will flock to this type of carbon source. Okay, remember petroleum hydrocarbon is yummy for bacteria that can break it down. And so there were indigenous microbes. And what they did was they added fertilizer directly to the site. That fertilizer supplied the needed nitrogen, phosphorus, and potassium so the organisms could thrive. They can't just live on crude oil alone. They have to get other nutrients, including micronutrients, which you can supply by fertilizer. The micronutrients would be in the salt water naturally, but not in a high enough amount in order to service degrading the plume. Okay, and so they was a researchers the crude oil, and they were able to for petroleum hydrocarbon. This was a different organism. It was metagen, was within the microbial, not just one microbe. And it was estimated that the half-life of the plume, the underwater plume, 3 days. So every 3 days, the oil plume would shrink in half. And when that happened, then it was very, very quickly absorbed with the real problem with the Deepwater Horizon was more this and also what each. So what they think is that this activity from oil fields below surface, and so they make this, and there is natural seepage oil about 25 on the floor. So already, there's natural selection here, even without the Deepwater Horizon spill. There's natural selection for oil degrading bacteria. So the scientists have for bacteria that were very deep in towards the ocean floor, and that they were migrating or grow um, when the hydrocarbon plume was released from the oil platform. Okay. So with this, there were about 900 different subfamilies of bacteria that were detected and newly discovered species of bacteria, obviously, all these degrading petroleum hydrocarbons. Okay. This is sort of an interesting figure. You look, here's a whale coming through here. Okay, but it shows some of the water contamination. It's out that the contamination minor until depth that was below the surface. Then you go up to this figure here, this insect, and you can see a droplet of oil, and then microscopically you see some rods that have migrated towards the oil droplet, and so they're being cultivated. And so you can see even more bacillus here, and you can see that there is some microbial activity that is naturally associated with the oil water interface. Okay. So the oil went into the ecosystem, especially in Louisiana, and so that did damage the shellfish industry, and the long-term impacts still have not been evaluated. British Petroleum was fined billions of dollars because of this oil spill and had to help foot the bill for the cleanup of the particular oil spill. And one of the hazards that came up was the use of chemical dispersants. These are chemicals like detergents that would break up the oil plume, and these were used, but they have some level of toxicity to the environment, so that had to be accounted for as well. In terms of future strategies, there is an ongoing effort to recover valuable metals by metal reducing bacteria, and these can convert microbes into metal oxides ores. And this is done for waste solutions for things like this boards. There are a lot of valuable metals, or a lot of waste that leaves the board facility will have valuable metals in it. So if you can reduce the metals, then that way it's covered. Radioactive waste. There are still over 100 sites that are contaminated by nuclear materials in the United States alone. And however, most radioactive materials still produce that type of ionizing radiation. But some strains of bacteria have demonstrated some potential for radioactive chemicals. But you cannot, it's very, very difficult to metabolize radioactive. There are bacteria that have the ability to survive radioactive contamination, but they can't take something radioactive material, and before that becomes non-radioactive, you have to wait time, regardless of whether it's bacteria or not. Okay. One of the things very recent was the Fukushima nuclear disaster due to tsunami that hit the and these are two videos. I'm not going to show you these videos directly on this video lecture, but I want you to take a look at them because the cleanup was so expensive. This isn't directly bioremediation, but it is important because it shows you the place. Again, regardless of whether you clean it up automatically or not, the radioactivity will persist for a long period of time. Okay, and that concludes chapter 9. We pause, and I'll come back with chapter 10. Okay, here is chapter 10, and it's all about aquatic biotechnology. And this is a growing field. There's a lot of interest in biotechnology, and there's a lot of aquatic biology in general because this is has been observed, and so there's a lot of research science that still needs to be developed in the area of marine biology and aquatic biology in general, and then to methods of biotechnology. Okay, so we'll introduce aquatic biotechnology, and then we'll talk about aquaculture. Aquaculture is not necessarily biotechnology, not yet, but it is used basically in order to create food, the world's food supply, namely aquatic species of food, using different biotechnological. And aquaculture is used prevalently for normal aquatic animals, but also with genetically modified fish. And we'll talk about genetic technologies and aquatic organisms. We'll talk about some of the medical applications where new devices and new materials being developed through aquatic biotechnology. Then we'll talk about non-medical products. And then we'll talk about environmental cleanup with the aid of aquatic biotechnology. Okay, so utilizing aquatic organisms could increase the world's food supply. It could restore and protect marine ecosystems. And we could also identify novel compounds for the medical field and also medical treatments. There are ways that we could improve the safety and quality of seafood. We need to be very careful with that. There's a lot of questions regarding consuming genetically modified seafood because that may introduce in and of itself. We can also discover and develop new products for chemistry. And so this is something that's outside the medical industry, it's something outside of food, but more is just polymers, chemical precursors. Okay, ways that we can monitor and treat disease. And then understanding, just increasing the knowledge of our biological processes and geochemical processes that are associated with the ocean. Again, this is sort of an untapped area of research because of the lack of accessibility of the majority of the ocean. Okay, and so we're going to talk about aquaculture. And we think of aquaculture just as being like a fish factory or any area where fish are being produced, or clams or mussels, any type of seafood that is being produced intentionally with intervention. So sometimes this is recreational, like stocking a lake, so fishermen catch fish, and then sometimes it's commercial for increasing the amount of biomass, where you have lots of fish through aquaculture methods that you wouldn't find in the wild. Okay. So the demand for aquaculture is expected to grow by 70% over the next 30 years. And there's a lot of problems with fish stocks. Their fish stocks are in open waters, and a lot of times there's treaties and there are negotiations between commercial fishermen, but a lot of times these are abused, and so fish stocks are being overexploited and depleted. And once they're overexploited, that basically that the fish stock needs significant intervention in order to. Okay, And to look in terms of aquaculture, this is where you've got human aquaculture provides 50% of all the worldwide fish. China is a leader in aquaculture. U.S. ranks about 13th. We do eat fish. We don't eat fish to the extent as they do in Far East Asia. And so you wouldn't expect that we would rank as high. And also our population is about a third of that of China. Okay, we're also looking at bait fish for commercial and recreational fishing, things like those. Different types of fish that are used in animal feed. Fish for culturing, mussels for culturing growth. Isolating pharmaceutical agents. And then ornamental fish that you would use like in a tropical fish tank you would buy at the store, or fish that are used for stocking for recreation. And if you look at aquaculture, there are a lot of different, we consume a lot of different types of fish. These are the primary organisms, both freshwater and marine organisms. Okay, and if you look at aquaculture, basically you have breeder fish that have eggs and sperm that are then cultivated together. These then develop to form embryos, and then you have small tanks where you have fish that are raised to a fingerling size. Then these, once they're reached about 3 to 6 inches, then afterwards on or raceways. Okay, and then once they reach a mature size for either market sale or for recreational fishing, then they can be released, or they can, and then they can be fished commercially, and so they're ready for market. Okay, and this just shows some of the, there's some raceways here and some aquatic ponds, and then you have areas here where nets, I think they're also abalone here, and it's out in the open water, but each one of these um, octagons, if you will, yeah, is enclosed by netting, and so the open water can flow through, but the abalone themselves are not able to get through the netting, and so they're able to get nutrition. You know, they basically feed by filtering water, and so they have this open discharge fresh water, but they're still encased so they cannot. Okay, in terms of fish farming, some of the innovations in West Virginia, there's a lot of abandoned coal mines, and these coal mines are being used to raise cold water species like trout or Arctic char, and you have to be very careful with this. You, if the fish are genetically modified, you don't want them to somehow get to the wild. So these are closed coal mines, and you have different types of culture. We call this, when you have more than one type of fish in a culture, we call that polyculture. And then there are also hydroponic systems where this is for recovery. Fish produce a lot of wastewater, and so if you want to recover the water, then you take the wastewater from aquaculture, subject to hydroponics, and then you have like flowers and vegetables, things like that, that are growing hydroponically that use the wastewater and treat wastewater, and then it can be recycled back when it's clean. So it's like a small wetland. Okay, in terms of improving strains for aquaculture, um, you want to look at different methods, both genetically and non-genetically, to increase growth rate, to control fat content. You don't want a lot of fatty fish with protein in it. And then obviously taste, texture, color. Okay. And so you're always looking at what the consumers want. And so if you can find a particular genotype that the consumers want, and you want to make sure you propagate that genotype. And then you can also diagnose disease. Aquaculture is contained, and when you have a contained system, then it's much easier for disease to spread than if the way disease is spread in the wild. And so if you're looking at different pathogens that infect fish, then you use different probes in PCR for in those parasites that infect fish, shellfish. Okay, some of the problems with aquaculture is that not all species are suitable for aquaculture. If you're looking at salmon, they have life cycles that are complicated and involve different types of water, and so that becomes more difficult because you have to have chambers and conditions for part of the life cycle. Like I said before, disease, when you have crowded and then you have contained conditions, especially with less genetic diversity, say if you have a monoculture fish, then it's very easy for disease to spread rapidly. And then excess consumption of wild caught bait fish. And so when you are consuming this type of bait fish, then you don't have anything to feed in aquaculture. And then pollution. Okay, fish farms, you know, if you've ever cultivated fish, if you've ever had a fish tank, the water gets dirty very quickly. You also have discharge from chemicals that are added, like antibiotics. And so you have discharge that's associated with those additives. And so these fish farms can be highly polluted, and their discharge then can harm adjacent aquifers. In terms of aquaculture, there's a use of biotechnology to promote growth and productivity, and also improve disease resistance and fish and shellfish. Okay, I think I will pause right there before we get into genetic technology. Okay, one of the biggest issues regarding genetic technologies is there is a lot yet to be known regarding aquatic organisms. And so just understanding genes involved in very basic processes for life are needed, especially things like survival under extreme conditions, say extreme cold or extreme pressure, and also learning about the immune systems, which are different in aquatic species. They do have an immune system, they do have blood, but there are some distinct differences in the immune system between humans and fish. New hormones that have been cloned include growth hormone. This is specifically for salmon. And then also antifreeze proteins, proteins that can be used to lower the freezing temperature. This is done in fish blood, and also extracellular fluids that cause fish to be resistant to freezing. And then also taking these genes and creating genetically modified fish, as well as genetically modified plants that are resistant to frost. And these antifreeze proteins, then you can also use these promoter sequences upstream of the antifreeze proteins in order to stimulate expression of different genes only during the cold. So if you have a promoter that is cold responsive, which the antifreeze protein gene promoter is, then you have something that can initiate different genes like reporter genes only under cold conditions. So you've got a switch, a promoter switch that turns off during heat and on during cold. Also, to use antifreeze proteins for cryoprotection of things like human cells, tissues, and organs. So these do need to, whoops, I'm going backwards. Let's see. Okay, other genes, glowing genes. There are a lot of marine um, organisms that glow, and so you have fluorescent genes like green fluorescent protein, bioluminescence genes. Remember, fluorescence genes require UV light, and bioluminescence genes basically take chemical energy and convert it to light. Okay, and these are good for different reporter genes, and so they allow detection of genes of interest. Okay, and here are some different reporter genes. This is green fluorescent protein, which is taken from jellyfish, and then that is uh, put as a reporter plasmid. And here in this particular document, and it shows the um, reporter plasmid then expressing in E. coli and NLC, and then it looks like muscles in panel B. I can't really tell. Okay. Also, looking at marine pathogens, understanding different things that infect marine organisms, the bacteria that infect salmon, P. salmonis, the genome was deciphered. We talked about this in microbiology, red tide, okay, which is caused by Pfiesteria piscidia, and this these genetics have been studied, and then other types of pathogens. I don't know, Pfiesteria piscidia. No, that's right. Okay, let me back up. Red tide is different. It's red tide is not toxic to shellfish. It's toxic to humans. But Pfiesteria piscidia is a different type of toxic. And this just looks at diagnostic techniques of different organisms, and you see some bands lining up specifically in oyster for this G. marinus, and it's just a diagnostic. They just basically are looking at PCR products for a particular toxin, and they use this to run PCR on organisms in a digestive tract of oyster, and they were able to identify different types. Okay. In terms of genetically engineered fish, there's lots and lots of them. There's only one on the market, and that is a type of salmon that we're going to talk about in just a minute. Generally, there's a lot of outcry against genetically modified fish, and a lot of companies have rejected selling these fish. Like Costco, you cannot get genetically modified salmon at Costco. Okay. And so they're looking at creating fish that grow faster and are healthier. And to make them faster and healthier, then they're looking at genetic modification, both to get them to grow quickly and as well to resist disease. One of the ways that they do this, while it's assuring that they will not overtake the wild type fish, is to create triplets. Triplets have 3 sets of chromosomes, not 2 sets of chromosomes, and they are genetically sterile. Okay. There are some instances where triplets can revert to diploids. So just by producing triploid fish doesn't mean that your line of fish is completely sterile when you do genetic engineering, because about 5% of the fish will revert to diploids. But it does provide some level of containment. Okay. Genetically modified salmon were approved in November 2015 for human consumption. These are called Aqua Bounty. Okay. And to look at some of the different things that have been done, a lot of these have antifreeze protein in them, AFP for Atlantic salmon, AFP, um, uh, that is AFP promoter um, with tied to salmon growth factors. So during cold conditions, they're producing more growth hormone. Um, other types of growth hormones. So um, there are some of these, like striped bass has insect disease, insect genes to prevent specific types of diseases. The insect being inoculated against the and so forth. And then down here, um, you don't think of rabbit as aquaculture. That's a little different. But what they're trying to do is um, genetically modify rabbit to produce salmon calcitonin. Calcitonin is a hormone that controls calcium loss from bone, and salmon calcitonin is already on the market um, as a bone loss prevention for patients that are older that are subject to osteoporosis. Okay, Aquabounty is the company, and they produce a transgenic salmon that grows 4 to 6 times faster than just wild type salmon. It's a cleanup salmon. And what they've done is they have stimulated higher production of the growth hormone GH, growth hormone gene. Okay. And they're using regulatory sequences from the Chinook salmon that these would be promoters that has more rapid growth on large vessel. And then regulatory sequences for the antifreeze protein from ocean pout. And ocean pout is just another type of fish. And by using the promoter, AFP promoter, um, associated with the growth hormone genes, and growth hormone is turned on during cold conditions, and that allows for growth during winter, whereas mostly wild type salmon just go dormant during winter and they don't grow as fast. Okay, so they reach the market size month and not 30 months, so they mature much more quickly, and so they can be harvested more quickly. Okay, here's some normal salmon on the top panel. You can see they're very small, and the Aqua Advantage salmon. The fish themselves are termed Aqua Advantage, and the company is called Aquabounty. Okay, so they are now approved by the FDA, and so Aquabounty does sell their salmon. But the main concern here is ecological impacts, because if the Aquabounty salmon ever got out into the environment, they would certainly outcompete wild salmon. Wild salmon could just not grow as fast, not be able to compete for food sources, and would obviously bigger salmon are going to have definite advantages, especially they grow so readily. So this could cause wild type salmon to be outcompeted, and we would lose the biodiversity of the wild type salmon. Okay, so these are female, and they're 99.8% sterile. That 0.2% still it kind of concerns me. The 99.8% are triplets. They have 3 sets of chromosomes instead of 2 sets of chromosomes, which renders them sterile. But still, if you have some that could slip out, then that 0.2% could end up posing an ecological risk. Okay. Also, there are genetically modified fish to glow. These are genetically modified strains of zebrafish, and they just have fluorescent protein genes from sea anemones. There are pink ones. That's the first genetically modified fish that was sold in the United States. This was before Aquabounty. Obviously, you don't eat GloFish; they're for tropical fish tank. Okay, And anti-biotechnology groups did not support this. And I think you have to be careful with any type of genetic modification. And so I understand the nature of those particular protests. I would be much more concerned regarding the Aqua Bounty company and their fish than with something like a GloFish, which is more of a novelty, just what GloFish look like. Now GloFish come in all different colors. You see kind of a red, pink, orange, and green. And then over here is the different species of fish. The ones on the left are zebrafish. The ones on the right, I'm not sure what those are. But these have green fluorescent protein. Okay, and you still need a UV light source for these fish in order for them to glow. They're not bioluminescent; they're fluorescent. Okay, polyploid species are created, triploid, and I just want to go into this um, further. So you subject fish eggs to conditions that interfere with cell division, and so the cells don't divide, but the chromosomes divide. And when that happens, they mature with an extra set of chromosomes. They grow more rapidly, but more importantly, they're sterile and they're triploids. Okay, so triploids, they're polyploids, and they look something like this. It's where the eggs are, the gametes are monoploid, and their growth is inhibited, so they go to triploid gametes that are fertilized with monoploid gametes. Okay, some of them might be haploid as well, depending on, you know, how. Okay, and diploid, then you could do a chemical treatment, and what happens in treatment is that you get um, a non-disjunction. Okay, so during meiosis, you get a non-disjunction here, disjunction, and when that occurs, you have, instead of two haploid cells, then you get one cell that's null, no chromosome, it's not going to survive. So diploid, it goes to meiosis two, it's still diploid, and then when a sperm fuses again, fuses with that, then it's a triploid. Another way to do this is to treat the egg chemically before it's fertilized, so it can accept multiple sperm, and when it accepts two sperm, then it becomes triploid. Okay. In terms of how these are regulated, primarily this is the National Oceanic and Atmospheric Administration, and this is through permitting. And so fisheries, marine sanctuaries, any type of protected species, when you're doing any type of aquaculture, you need permits for that. And then if you do any type of scientific research or you start to genetically modify marine organisms, then you need a scientific research enhancement permit. Okay. This is not correct. Okay, this is I still haven't fixed this slide because Aqua Advantage has been approved. It was approved in 2015, and preliminary findings of the FDA were positive, and obviously they accepted it for consumption. Okay. Now, the FDA required that only 95% of the fish produced be sterile. Okay, so 5% were fertile, which again, that means that these genetically modified fish could outbreed wild type fish within 40 to 60 generations. And the GM fish all have the same genotype. They're all basically clones of each other. And so when you have these clones, then these could outbreed wild type fish. And when you outbreed wild type fish, the aqua advantage fish then are going to limit genetic diversity. Okay, you know, once the wild species diversity dies off, then you have just a single species of genetically modified fish. Okay, this could spread to wild populations, and it should be said that one of the stipulations of the FDA is that salmon has to be in landlocked farms. Okay, so it cannot be the reservoirs where they grow can't be in any type of natural streams or natural lakes or flow out in rivers or oceans, and so they're completely landlocked. So to prevent contamination, making sure that these um, don't get into the wild. But they could leak into the environment, and if they leak into the environment, then they could spread diseases that maybe aquatic but we don't know how the wild population is going to react. We also have to look at potential human health effects of aqua advantage salmon. You have to realize that this fish has more growth hormone in it. Okay, so it has higher salmon growth hormone levels than normal fish. And so growth hormones, salmon growth hormones may be reactive to human tissues. There is a lot of sequence similarity between the two types of growth hormones. And this, just by having growth hormone, this could lead to rapid, undifferentiated growth of human tissues, and it could lead to specific types of cancers. Also, allergic reactions to genetically modified salmon have to be taken into account. And then antibiotic use, because they do use a lot of antibiotics in aquaculture. And so the more that the fish are exposed to antibiotics, the more that people that eat the fish will also be exposed to antibiotics. Okay. And another thing that I take issue with, and this is the way the Food and Drug Administration works, is that the company who is seeking approval for, in this case, the consumption of genetically modified fish, AquaBounty is actually submitting all of the data. They supply all the data that's reviewed by the FDA. So there is no independent verification. Only AquaBounty employees are generating these data, or somebody who is hired by AquaBounty to do independent confirmation. The FDA does not do the testing of the fish directly. And so I kind of take issue with that. I wish our FDA could do more independent testing on their own so they would have some type of unbiased opinion of how these salmon are going to act in human populations. Okay. Now let's just change subjects to medical biotechnology. This is very important because there's a real new frontier in terms of different drugs that can be developed through marine biotechnology. This includes bioprospecting to get different types of medicines. One of them is calcitonin. Calcitonin is a salmon product. It's a hormone that delivers calcium to the bones, and it's 20 times more active than human calcitonin. So you only need small quantities of this. So if you want to enhance the amount of calcium that's taken up by the bones to prevent osteoporosis, then you can prescribe salmon calcitonin, and it is already on the market. Okay. Another important product is something called hydroxyapatite, and hydroxyapatite can be derived from the bone of marine organisms, and this is comprises an implant. And these implants then can be used as like bone paste and fill the gaps in fractured bones. Okay, byssal fibers. Byssal fibers are the fibers that mussels use to attach to surfaces like boats or rocks or anything. And you know that mussels, when they attach to they're very difficult to get them to detach. Okay, and these byssal fibers then make really, really great adhesives like surgical adhesives. Okay, here's salmon calcitonin. This is a nasal spray. Okay. And these are byssal fibers that you can see the individual fibers where the mussel is being pulled away from the surface, so the fibers are tight. Also, anti-inflammatory compounds, analgesics. Analgesics are things like ibuprofen or Tylenol, and anti-cancer compounds. Okay. Conotoxins are being used, which is a type of drug that is a painkiller. Yondelis is an anti-tumor drug. It's a chemotherapy drug, and that is from the sea squirt, and it binds the minor groove of DNA and inhibits cell division. And you want to block cell division to block tumor growth, and that blocks transcription and DNA repair. Okay, and here are some other medical products that come from other organisms. Leeches. Leeches prevent blood from clotting. Okay, and they produce a compound called pyridine. And pyridine is an anticoagulant that is used to, if somebody has a problem, then pyridine will prevent the blood clotting. And there are some other therapy drugs here, anti-angiogenic, antimicrobial, and so forth. Okay, the pufferfish produces a nerve agent. I don't know why this would be a tetrodotoxin. I don't know why that would be considered a medicine if it's more lethal than cyanide. Okay. Oh, here we go. This is to better understand how sodium channels help neurons produce electrical impulses. And this led to the development of new drugs being tested for anesthetic and anti-cancer agents. So TTX is not a drug itself, but it's used in drug development. Okay, here's some pufferfish. You can see them, they're not quite puffed up, but these are used to make TTX. Other types of non-medical products include enzymes. These are enzymes that you would use the first three DNA polymerase, ligase, and restriction enzymes for molecular biology. And then proteases and collagenases you would use in other applications. Carrageenan is it's a type of gel agent. It's similar to like agar. It's used in processed food, and you also get carrageenan paste, and it's used in toothpaste. Biomass. Waste biomass, and can be harvested to make energy. And so this would be any type of biomass that can be converted to glucose and then fermented to ethanol. So that would be seaweed, any type of grass from the bottom of a marine environment or plankton. And then algae has also been used as sort of recombinant platform for genetic modification. Anti-fouling agents, these are biofilms preventing agents. So different agents that will prevent barnacles, algae, mussels, clams, and bacterial biofilms are also found in the marine environment. Okay, and this is just a type of biofouling. You can see the mussels are growing there on the shoreline. Okay, and here is an anti-fouling agent here that is trying to keep itself from being biofouling, and it's secreting a compound that keeps these this other organism. I'm not sure what this is, bacteria, keeps this other organism. Okay, so you've kind of got to. Okay, and I think I will leave off at this particular point, and we'll take up the rest of this on Monday. No, actually, there's only one chapter. Okay, so anti-fouling agents. Traditionally, these were toxic chemicals, things like copper-rich and mercury-rich paints that you don't want in the marine environment. So now they're looking for natural anti-fouling agents for marine organisms that protect their own surfaces. Okay. Also biosensors, things like Vibrio. This should say Vibrio, it says Vibro, but Vibrio bacteria are bioluminescent, and they have been used. You can take Vibrio genes and put them downstream of promoters that are sensitive to different pollutants, and you just look at the light intensity of the bacteria, and that will tell you whether pollutants are present or absent. Okay. And then also environmental remediation, which we talked about in chapter 9. And that concludes chapter 10 and also concludes our lecture for today. So we'll pick this up on Monday after spring break.